Okay, in the first two parts of this unit, we've focused on uh, how the cell membrane works, how water continuously flows in and out of the cell membrane to attempt to maintain equilibrium with the cell's external environment. And then we looked at the two types of cell diffusion, active or passive transport and active transport, and we focused mainly so far on passive transport. So in this part, we're going to be getting into active transport. Remember, passive transport doesn't need energy to move molecules across the cell membrane. Active transport, however, does require energies to move molecules across the cell membrane, because what you're doing is you're moving those molecules against the concentration gradient. Remember, concentration gradient, we just looked at this. Usually through diffusion and passive transport, you can have molecules that are highly concentrated in one area, and they naturally spread from that higher concentrated area to a lower concentrated area to balance out. Okay? Doesn't take energy. That's natural progression. In active transport, what you're doing is you're moving molecules against that concentration gradient. So take for example our pizza smell example. You're taking the smell of the pizza that you can smell all the way in the back of the room, and you're pushing all of that smell all the way back up to the front of the room where the pizza box is, so where you can't smell it anywhere else except where the pizza box is. That's moving against the concentration gradient. That requires energy from the cell. And it also requires proteins that act as pumps to move those things physically using energy. They pump them against that gradient. A very common type of protein pump is called a protein ion pump. Okay, in this particular example, you can see these. Remember, we talked about ions, positively charged molecules. In this example, you can see that there are positively charged hydrogens or hydrogen ions on the inside. Okay, here's the cytoplasm. So this is the inside of the cell. Notice that there's a higher concentration of hydrogen ions outside the cell. But what's happening is the cell, for whatever reason, needs to remove excess hydrogen ions, so it is pumping it through this protein pump. It has to add ATP, or it has to add energy to do this, to move this hydrogen out of the cell to the outside of the cell. It is moving it against that gradient because naturally these ions would move inside the cell, but the cell doesn't want hydrogen, so it is pumping them out of the cell against the gradient. That requires energy. Another type of pump is called the sodium-potassium pump. This is a very common type of protein pump used by the cell as well. Pretty straightforward. What happens is sodium is going to be pumped out of the cell, and the cell is going to swap the sodium with the potassium and bring the potassium in. Okay, both of them are going to be pumped against the concentration gradient, which means you're moving sodium out of the cell to a higher concentrated area, and you're bringing extra potassium into the cell against the concentration gradient. The cell needs potassium. It can swap potassium and sodium very easily through the sodium-potassium pump. It doesn't want excess sodium, so it gets rid of it. It wants to bring in extra potassium for processes, so it brings it in, but it's going to move it against the concentration gradient, and it's going to use that sodium-potassium pump. Here's a nice illustration of how it works. Okay, the cell has too much, or has sodium, but it doesn't want it. There might be excess sodium outside of the cell. So what it's going to have to do is it's going to add sodium to the sodium-potassium pump. It's also going to add energy to the pump to make this process work. It's going to pump that sodium out of the cell and then swap it with potassium. Now there's going to already be a lot of potassium inside the cell, but the cell knows that it uses a lot of potassium for a lot of different processes, so it's going to bring extra potassium in at the same time. When it does that, it's going to require that energy. The potassium comes in essence, that's how it works. Okay, a similar example to what we looked at with the hydrogen pump. And once again, this is needed. Energy is needed to do this because there's already, for example, potassium. There's already a lot of potassium inside the cell, but it's trying to bring more in for other processes. There might be a lot of sodium outside the cell, but the cell's trying to get rid of sodium. So it's pushing it against that gradient It's going to require energy. Alright, good little picture. Okay, the top part is letting you see passive transport. So you notice over here, you've got just regular diffusion. Remember, water is moving across this membrane all the time to try to maintain balance. Water is moving through osmosis across the membrane. If you have a higher concentration of molecules outside, the cell needs those molecules, or if there's just a higher concentration of molecules, the cell might not need them, but there's a higher concentration, it can move against that gradient. It can move, or it can move with the gradient. It'll flow from higher concentration of molecules to lower concentration of molecules across the cell. That's diffusion. Okay, the molecules are small enough, dissolved in water, to go ahead and pass through the cell membrane because of the flow of water. So that's just plain old diffusion, passive transport. Here, these molecules are a little too big to pass naturally through the cell membrane. However, they're still going to move from a higher concentration to a lower concentration to the inside of the cell, but they're going to flow through this protein channel. That's facilitated diffusion because the protein channel is going to help facilitate the diffusion process. Doesn't require energy, that's passive transport. Here, the cell has a large quantity of molecules already inside its cell. It wants more because it knows it's going to need to use them, so it's going to add energy to this ion pump, this protein pump, and it's going to bring in more molecules against the gradient. So they're going from low concentration to high concentration. That is against the concentration gradient. It's going to add energy to that protein pump, and then you have active transport as a result. So that's a good picture to focus on. Alright, let's look at movement of these molecules. Molecules usually move around in the cell, in and out of or to be removed out of the cell or get engulfed into the cell by vesicles. We talked about what vesicles are. Now, when vesicles are going to be used, you're going to have these large amounts of substances that are just way too big to be moved through protein pumps, protein channels, or to be moved through osmosis. So they're going to be transported by vesicles to the membrane or from the membrane into the cell. Okay, for example, proteins are way too big to flow through a protein channel or flow through the cell by osmosis. Polysaccharides, same thing, way too big. So vesicles are going to be used to move those types of examples of substances in and out of the cell. Alright, how it works, the vesicle kind of engulfs that substance. It's going to form a pouch around, say, the polysaccharide or the protein. Okay, it's going to pinch itself off, forming, in essence, forming a cocoon or forming a bubble around that substance, and then it's going to transport it. If the vesicle is going to move the substance into the cell, that is called endocytosis. If it's going to remove a substance like a polysaccharide or protein out of the cell, and remember these are molecules that are too large to pass through the membrane, okay, using a protein pump or using osmosis. If it's going to move things into the cell or out of the cell using vesicles, into the cell is endocytosis, out of the cell is exocytosis. What does cytosis mean? We've kind of just covered that. All right, we looked at what endocytosis was and what exocytosis is. Most of the time, exocytosis is going to occur when you have a large amount of waste that needs to be packaged up and removed. The Golgi, remember we talked about the Golgi. It's going to be used for packaging and shipping. So the Golgi is going to package this stuff up. It's going to form these vesicles. It's going to transport these vesicles to the cell membrane, and then it's going to remove them from the membrane. Here's a nice picture. You can see the vesicle moving to the cell membrane. It fuses with the cell membrane. Then it opens up and it removes the waste. Here's another picture, an example. Vesicle carrying waste, transporting it to the cell membrane. It fuses with the cell membrane, and then it allows the materials to be excavated from the cell membrane or released from the cell membrane. Endocytosis, bringing large things like maybe extra polysaccharides or extra proteins, something like that, into the cell. Here we go. We have these large bundle of materials. The the cell is going to make a vesicle from the membrane itself. It's going to encompass those particles that it wants to bring inside the cell. It's going to pinch itself off, and it's going to travel down to the Golgi, where the Golgi can help transport endocytosis. Now, there's a third type called pinocytosis. This is a type of endocytosis, but this pinocytosis is where the cell is kind of drinking in extra liquid into the cell. But it does it in the same process. Okay, phagocytosis or phagocytosis is where the cell is going to actually engulf large food particles and bring it into the cell. So pinocytosis is bringing in liquid through endocytosis. Phagocytosis is bringing in large food particles through endocytosis. Protists use these two processes very easily, very efficiently. Bacteria do the same thing. And then here's a nice process of phagocytosis. The cell is going to bring those particles in. It's going to use what it needs. It's going to get rid of it. Okay, very straightforward. Here's a white blood cell encompassing a bacteria. It's going to eat it, break it down. All right, then the last thing of type of endocytosis, this receptor-mediated endocytosis, is very specific, and it's not used very often, but it is very, very specific to, remember there are those receptors, these carbohydrate glycolipid receptors on the outside of the cell. They're kind of like antennas, all right, and what they do is they are triggered by specific receptors of specific molecules outside the cell to cause endocytosis to occur. Here's a good little picture. You got the Golgi. You see exocytosis happening on the top part of the picture. You see the endocytosis on the bottom part of the picture.